the overall strategy to reducing the number of people who face a cervical cancer diagnosis does really require thinking about all of those pieces: vaccination, screening, and treatment.
This is the Discovery Files podcast from the U.S. National Science Foundation. I'm Nate Podker. For millions of people around the world, healthcare is not a regular occurrence, and in developing countries, it may not be available at all. Our guest today, Kathryn Kundrod, is working to address global health disparities. While earning her PhD in bioengineering at Rice University, she helped develop an integrated HPV DNA screening platform that could revolutionize cervical cancer screening and cost less than $5 a test. Today, Dr. Kundrod is in the Cancer Prevention Fellowship program at the National Cancer Institute and contributing to the White House Cancer Moonshot. Thank you for joining us today.
Thanks for having me.
So I want to start with asking you about what drew you to working with public health issues. You've done a lot of work with HIV testing and cancer testing.
Yeah, I've long been interested in social justice, equity, thinking about societal inequalities. And when I was studying bioengineering, I became very interested in the role of diagnostics and screening tests and kind of different access that people had to having that health information to help direct their next steps medically. And so I became very interested in point of care diagnostics as a tool to increase access, bring testing closer to people where they live. And both HIV and cervical cancer really drew my interest because they're two situations in which we have a lot of scientific knowledge now about how to effectively prevent and treat both HIV and cervical cancer. And really, it's a question of access to medications, diagnostics, and treatment that keep the both diseases prevalent. And so that's been how I got involved in both HIV and cervical cancer, really from the bioengineering perspective and thinking about access to screening and diagnostic tests.
So thinking about that access, what are the biggest challenges in deploying this kind of screening in low and middle income countries?
I would say the two biggest challenges fall into cost and complexity buckets. So cost being a really major barrier right off the top. Most tests, the instruments cost kind of tens of thousands of dollars. In a lot of cases, the per test cost can be really high, and that's just prohibitive in a lot of low and middle income country laboratory testing scenarios. And then complexity, which is what I think about a lot as an engineer. So thinking both about complexity of use, so really designing tests well, human-centered design to be sure that the test is really intuitive for a laboratory technician, and then also thinking about the complexity of the instrumentation. These tests need to be robust to temperature, humidity, electricity fluctuations, and environmental conditions, especially in open air hospitals. So keeping instruments in use, and then when they break down, having the knowledge and spare parts available to be able to fix them. So really thinking about that from the beginning when you're designing the instrument to be able to withstand conditions and have a path forward where spare parts aren't so hard to get that that a repair is really challenging and kind of renders the test useless in that environment.
Right. And this really feeds into what we're talking about today with your integrated test. Can you kind of give us an overview of how the finished test works?
Yes. First, a swab is collected. It can either be collected by a physician, and that would be a cervical swab, or by a patient themselves, and that would be a vaginal swab. You then place the swab into a liquid that breaks open the cells, releases DNA, and that's what ends up being tested. You do a little bit more processing of the sample to kind of isolate that DNA and get rid of the other parts of the cells that could get in the way of the testing process. And then you add that sample to pre-dried materials that can make copies of that DNA. So it's very similar to a PCR test that has become really routine in this COVID era. but does so at a single temperature. So there's some extra enzymes in there that can break open the DNA and make a copy of it. And then that process is referred to as isothermal amplification. So you're amplifying the DNA to detectable levels. Then within an enclosed system, which is important for not releasing that DNA into the environment, which can then cause false positives on future tests, we transfer that amplified DNA to what's called a lateral flow cartridge, which I think, again, in recent years has become something that a lot of people are familiar with rapid COVID tests. And the benefit of a lateral flow cartridge is it uses paper to bring the sample down past kind of a test area where in the presence of amplified DNA, you get a color change. You get a pink line or blue line showing up that indicates that the thing that you're looking for is there, in this case, HPV DNA. Or in the absence of that line, you know that it's negative. So what that paper test allows you to do is avoid kind of robotic arms to move samples around within an instrument or things like that, speaking back to that piece of instrument complexity. So from sample to answer, you have the swab going through that DNA amplification process within a cartridge, transferring into that paper-based system, and then providing a visual answer on whether or not there's HPV DNA in the sample.
Right. Not a lot of steps compared to other kinds of testing that might be wider spread. You mentioned screening specifically for HPV when approaching cervical cancer. Why is that the best way to screen?
Yeah, nearly every case of cervical cancer is caused by an HPV infection. We now know that it is the necessary cause of cervical cancer. So when you're looking at a whole population, you might catch a lot of HPV infections that aren't progressing to cervical cancer, but you also know that this is the group of people that you need to kind of watch more closely because they have the necessary cause. Mm-hmm. So it in terms of test performance, it's a very sensitive test. You catch all of the people who are at risk of developing cervical cancer, which is really important for screening. There are other tests that have really good quality evidence behind them, like Pap tests or cytology is the other term for those, which are in very widespread use in the U.S. And work really well, have really brought down cervical cancer screening or cervical cancer rates in the United States and in other high-income countries. The challenge with cytology is it requires a trained cytologist and different instrumentation, whereas molecular tests, like an HPV test where you're looking for DNA, can be developed into these smaller devices and can be made in formats that can be more decentralized and more accessible.
So can you talk a little bit about the test itself? I want to really get at the benefits of using the disposable cartridge system and kind of what the costs would be for an open air hospital, let's say.
Mm-hmm. Yeah. So one of the main benefits I sort of touched on already, which is the cartridge encloses the test. So that prevents what's referred to as environmental contamination, where if you do release DNA into the environment, that can be a huge challenge for downstream testing. Using a disposable cartridge, in addition, depending on how it's designed, in this case, it can be produced with injection molding. So you can produce a lot of cartridges pretty readily with one injection mold setup, and reduces the per test cost when you can just kind of continue to manufacture those. In addition, it really reduces the instrumentation complexity. So as we talked about, there aren't really robotic parts to the instrument, so there's less chance that it breaks down. And effectively, what the instrumentation is for this test has two parts. One is a 2-temperature heat block, one that is used for breaking the cells open, and one that's used for amplifying DNA. And then the other piece of instrumentation is used for preparing samples in that sample preparation protocol where we're kind of separating the DNA from the rest of the cellular components. And that's just readily available, kind of off the shelf. It's a benchtop centrifuge. So the disposable cartridge does a lot of the work that instruments often do, and does so at a lower cost.
So it'd really make it a lot more accessible in general.
Exactly. So bringing down the cost can allow for more clinical settings to afford it, and can allow for, with that simple instrumentation, can allow for kind of smaller clinics outside of centralized labs, thinking about things like mobile testing scenarios. And to your question about cost, we're still estimating cost at this point, and we won't know full costing numbers until a little later in the process. But we are estimating, based on the cost of the reagents and materials, that we would be able to achieve less than $5 per test, $5.
That's pretty crazy. As you were working on the project, what were the biggest challenges along the way for you?
The biggest challenges, I think, on the technical side, had to do with the fact that samples collected in traditional cervical cancer screening programs really aren't very compatible with point-of-care testing. So thinking about the samples that are needed for a pap test or cytology, you really need to preserve the cell and keep it intact, which is kind of the opposite goal of when you're looking for DNA. Breaking those cells open and getting to the DNA becomes harder when you kind of really preserve the cells well, which is the goal of most of the collection solutions that we use in standard programs. So sorting through that sample preparation protocol was a major technical challenge. And then the logistics of designing a test well for use in low and middle income countries, getting the materials to a low income setting. I had the fortune of working with really amazing collaborators in Mozambique and El Salvador through my graduate training while I was developing this test, and making sure that I got good user feedback in the settings where these tests were intended to be used, required a lot of logistics and bringing all of the materials to people and incorporating their feedback. So both on the technical and logistical side, I think there were challenges, but we had such an amazing team supporting the work that we could have faced a lot more challenges had we not had such brilliant minds on the job.
So on the other hand, what was the biggest surprise?
I think that I don't know that it was a surprise necessarily. I think we expected performance to look a little bit different outside of the lab that we developed it in, the test in, versus in kind of field use. But in the study that we just published, we saw that when we were testing in Houston, in the lab that the test was developed in a high resource setting with really controlled conditions, we had really good performance of the test. We basically detected every sample that we would have expected to. And then when we took the test to Mozambique, we didn't know what challenges we would face in that scenario. And so we definitely went in expecting to learn. And I think the thing that we didn't expect there was, instead of missing just samples that had like a really low amount of DNA, we also missed some that had higher amounts of DNA. And we learned that one major difference between these two settings was in Houston, physicians collected the samples. In Mozambique, patients collected their own samples. And some of the samples that we missed there, some of the positives that we missed, had really high numbers of cells in them. And so we looked into that and learned that self-collected samples have a higher cellular content than physician-collected samples, and you can get to a point where it's too high, and it just blocks the downstream process. So that was a really important learning, because those are the samples that would allow for truly like mobile testing. Somebody collects their own sample and puts them in tests. So we didn't expect that, but it informed what needs to happen next, which is really making sure that we understand if we miss a sample, was it because of too high a number of cells in the sample, or was it because really there was no HPV DNA? So that will drive forward the next step of development.
So I believe while you were working on this, you received a Graduate Research Fellowship grant. Can you talk a little bit about your experience with that program and what could you have only done with that award?
Yes. I was very fortunate to receive a Graduate Research Fellowship while I was a graduate student at Rice University, and it was a fantastic experience. I think putting together the proposal was one of my first experiences of really thinking about what I wanted to do with this period of training. And receiving the fellowship gave me a lot more flexibility to answer the questions that I thought were most important to my graduate training and to the big questions around kind of diagnostics access that I wanted to improve. And so what I wrote my proposal on was on sample preparation for point of care diagnostic tests, which is, I think, a major challenge and is really hard to present as kind of the thing to focus on. Thinking about those amplification and detection pieces in point of care diagnostics really are much more tangible, much easier to understand. But I knew that in order for a test like this to work, we really needed to get the sample preparation piece right. So it really afforded me that flexibility to be able to work with our team at Rice and beyond to prioritize the sample preparation piece of point of care testing.
Awesome. So I know you got your doctorate now and you've moved away from Rice University. What is the next step for you personally, and what's the next step for your test?
So for me, I joined the National Cancer Institute's Cancer Prevention Fellowship Program following my PhD and postdoc at Rice University. The reason I joined this program was I wanted to focus a little more on the public health pieces of this work. And so through that program, I got a master's in public health, and now I'm working on public health-focused cervical cancer prevention research at the National Cancer Institute. So that's what my next steps involve. I'm thinking about things like the technical barriers around HPV testing still. I'm thinking about what program managers might need to know in order to effectively implement HPV testing and staying connected to that world, but thinking about kind of the public health program level of cervical cancer prevention. And the next steps for the test are, I think there are three main things that need to happen. One is this test detects HPV 16 and 18. Those are two types out of approximately 12 that are known to cause cervical cancer. They cause the vast majority, over 70% of cervical cancer cases are caused by these two types. So it's really important to detect them, but I don't think they're enough on their own. So one technical next step is to incorporate maybe three more types. The second piece is addressing that challenge on sample preparation, where if there are too many cells, they get in the way. And then the third is just wider spread testing. We need to make sure that this test works in diverse populations, in diverse laboratory settings, and really provides very clinically useful answers.
Special thanks to Kathryn Kundrod. For the Discovery Files, I'm Nate Podker. Discover how the U.S. National Science Foundation is advancing research at nsf.gov.